So let's do some classification of epithelia and get into the real nitty gritty here of it. And there are two ways to classify epithelia. The first is how is it layered? And one way that epithelia can be layered is just to have a single layer of epithelial cells. Remember, epithelia is lining things. So below the epithelial tissue, we'll have other tissue types. We'll have the basement membrane, of course, just below it, but below the basement membrane will be connective tissue probably. Above the epithelia, there won't be anything. So we have a single layer of cells in between the deeper connective tissues and the opening in the tube or whatever we may be encountering the epithelia. Now, besides this simple layering, we can have multiple layers. And if we have multiple layers of cells, those are stratified. So stratified is anything above a single layer, all the way up to 20 to 25 layers of cells. And lastly, there's kind of an in-between one. It looks stratified because of the way the nuclei are placed in it. And we'll see plenty of examples coming up here. I also encourage you to look at all the examples in your APR that I'm going to be posting and so forth, just to kind of get a good feel on how am I going to be able to tell one tissue type from another. And so this third type, pseudostratified, looks like it has multiple layers because nuclei are placed all over the area. But the definition of being a simple layer is that all cells above the basement membrane maintain contact with the basement membrane. Stratified won't have that. You'll have a layer of cells that contact the basement membrane, but each subsequent layer is above that base layer of cells, so they don't contact the basement membrane. But here, every cell does in pseudostratified. So it is pure stratified, but it is actually a simple layering of cells. Now, the other way we can classify epithelial tissue is by cell shape. And in this regard, we can have irregularly shaped cells that are called squamous. We can have cells that appear squarish. They are called cuboidal. And then the cell types that appear rectangular. Now, we can start to combine all of these terms, this layering along with the cell shape. And so we can get simple squamous, simple cuboidal, simple columnar. We can get stratified squamous, stratified cuboidal, and stratified columnar. Pseudostratified, fortunately, though, only comes in one flavor, and that is as a columnar type of cell. So you'll only hear about pseudostratified columnar. Now, each of these are ideally suited for what purpose and their location in the body. So let's get to that. I mean, that's kind of the neat stuff about all of this. So here we have some pictures showing you the different ways that epithelial tissues can be classified and arranged. And so let's begin by looking at a specific examples here of simple squamous. And simple squamous cells, this is great pictures. I love these pictures. The left one is a real photomicrograph. The right one is somebody hand drawing it and illustrating it and then labeling it. And so I think they're both really useful to help you start to identify the important features of these particular tissues. And you see the white space that is above the layer that we're looking at, the simple squamous layer. This could be lining a blood vessel. This could be lining a tubule of some type. All right, so common functions here are that we'll get diffusion and transport across these very thin layers. It makes sense. You probably don't want to provide much of an impediment to diffusion, and so you need this very simple, short, thin, not short, but thin layering. And we find these in many, many locations in the body. Simple cuboidal. Simple cuboidal, again, beautifully illustrated here. You see it. This is a kidney tubule. That's the most common location for simple cuboidal. I want you to know locations for each of these tissue types. Simple squamous, lining blood vessels. Simple cuboidal, kidney tubule linings. And they are primarily there for absorption, secretion, since we are talking about the kidneys. You should also know about one function that they might have as well, but in particular, definitely know their location. The third type is simple columnar. And in simple columnar, you can have two different types, and one can be ciliated or it can be non-ciliated. Typically, you'll probably see pictures of ciliated simple columnar tissues. Very commonly found in the digestive system. And I would say that this picture is from the tubules of the small intestine. And you see here the nuclei are all nicely, neatly arranged, pretty much at the same level in the cell. You also see this clumping of what kind of looks like maybe some adipose, some fat. But this is a simple gland called a goblet cell. And it's pretty much always associated with the simple columnar tissue type in the digestive system, as well as pseudostratified columnar found in the respiratory system. And goblet cells produce mucus, tons of mucus. And so you'll commonly see them. Since we're going to be talking about glands, you might as well start to get used to goblet cells quite a bit. So we're going to be talking, since in the digestive system, a lot of absorption going on here. Finally, the pseudostratified. So hopefully you can start to see exactly what I mean here by pseudostratified. And we'll always see it as ciliated. So pseudostratified, we'll always see it as ciliated. We'll see it associated with goblet cells as well. But on the illustration especially, you can see that. Now, it may not look like every cell is contacting the basement membrane, but it's just that some of them are being obscured by other cells, but each of them will definitely contact the basement membrane. But you can see how this multiple layering of nuclei almost makes it look like it is stratified. So again, commonly found in the respiratory tract, and we'll see it associated with goblet cells, so there'll be lots of mucus in association with it. So that covers our simple types of epithelium. Let's look at stratified types. And so here we have a simple layer, or a single layer, that's going to be resting against the basement membrane, but each of the other layers are above that single layer and don't make contact with the basement membrane. So we have the same three types: stratified squamous, stratified cuboidal, stratified columnar. But you'll see strike-throughs on the cuboidal and columnar, indicating that they're really minor in their distribution in the human body, pretty darn rare. And so we're not going to really worry about them at all. We're not pathologists. We're not going to worry about the cell types or cancer development in them or anything like that. So we're going to focus on stratified squamous because it happens to be what makes up our skin. Now, that's not the only location for stratified squamous. It is found in many other places, and we'll talk about that. There is another type of stratified tissue called transitional. So we'll discuss that. It's found in a particular location in the body as well. So stratified epithelia, this happens to be a type that's called keratinized. So it's full of a protein called keratin, which helps to seal it. Thus, I would say this is skin, and indeed you can see this particular examples from the bottom of the foot. And we can see how there are active cells, these ones kind of stained with purple, and below kind of looking like fingers extending into the tissue deep to it. And then on the surface, you'll see this layer of cells that doesn't appear to have any nuclei, anything active going on in them. And indeed, they actually are dead. They are full of keratin. And so this is this crusty layer that waterproofs the skin and prevents leakage or entry of fluids inappropriately. Well, if we have keratinized stratified squamous, obviously we must have non-keratinized stratified squamous. And this particular example is in the vagina, but you certainly have other examples in the oral cavity and such. And we can see here we don't have that layer. Let me bounce back again. See that layer of keratinized that's extending towards the left of both of these figures. And then here you just have a mass of active living cells for the most part. So that's stratified squamous. Again, skin found in the esophagus because we put lots of rough stuff down our digestive tract, so it's not quite moistened often too much before it hits the esophagus, and it can rip and tear cells. So it makes sense to have multiple layers of cells that can immediately take on the role of protecting the esophagus. In our oral cavity, we have lots of stratified squamous, and down in the anus as well. I'm sure you realize that things can get rough down there as well. How about transitional epithelia? Transitional epithelia is unique to the bladder and to some extent the urethra as well. The ureter. And so transitional epithelia looks very similar to the stratified squamous. But on a lab practical or a lab quiz, I would caution you, try to figure out features here that clearly demonstrate a difference between transitional epithelia and stratified squamous. Note the roundness of the cells in the transitional epithelia. Go back here, look at the flattened cells in the stratified squamous. They're not round at all, especially as you get near the edge, near the white space. And here, certainly very much flattened. But in the transitional epithelia, we see very much rounded. Now, what is the goal here? What is the role of this epithelia being in the bladder? Well, what does the bladder do? The bladder fills with urine and expands, and then you drain the urine and it collapses, right? Well, we need an epithelia that lines this structure that can adapt to this increase and decrease in size that alternates a few times a day. And so as the bladder fills, you'll see this transitional epithelium thin out. So it may be this thick, and then as it fills, it thins out. Then as the urine empties, it will begin to thicken again. So we have this capability, this unique capability, really impressive, of thinning and thickening depending on the state of the bladder.